Here we go. We're gonna go over the warm-up, look at a little review of the endocrine, and then we'll do our last topic of the semester, which is gonna be going into the reproductive system, which we've already kind of introduced through discussing gonadotropin releasing hormone and FSH and LH and some of the basic things going on with those, except we're going to look at all that in way more detail. So let me share screen here. And it says, which of the following glands release hormones involved with regulating calcium levels? Most people got parathyroid. Those were the ones that released the hormone that, what conditions are going to cause the release of parathyroid hormone?
Low calcium.
Yeah, low calcium. So if your calcium is low, your body needs more calcium. Parathyroid hormone is released, which is going to release some of your stored calcium from your bones and bring your calcium levels back up. The thyroid gland releases calcitonin, which seems to be more about like kind of storing away calcium, but doesn't seem physiologically as important in terms of just everyday functioning as the parathyroid hormone. Name a specific hormone that controls the release of another. Most people were getting this. So again, tropic hormones are really important and common, like thyroid stimulating hormone releasing thyroid hormone from the thyroid gland, adrenocorticotropic hormone releasing cortisol from the adrenal cortex. We also talked about a lot of the releasing hormones from the hypothalamus, like gonadotropin releasing hormone causing the release of LH and FSH from the pituitary. And then like LH from the pituitary was causing the release of testosterone from the testes. So there's lots of different answers there that would be the right answer. And then we also talked about there's often kind of negative feedback going on. We'll see more of that today as well when we look at the control or the kind of cycling and control of levels of sex hormones in your body. Match the hormone with the gland that produces that hormone. Calcitonin, most people got was thyroid. Epinephrine, again, adrenal medulla. The middle inner part is epinephrine or adrenaline. The outer part is making the steroids like aldosterone and cortisol, as well as some sex hormones. Insulin, everybody got was a pancreas. Oxytocin, is posterior pituitary. Posterior pituitary's got oxytocin as well as ADH. And all the other pituitary ones are going to be anterior pituitary, such as growth hormone in the anterior pituitary. Aldosterone, again, adrenal cortex. Thyroxine. And again, we talked about thyroxine, being T3, T4, the triiodo and tetraiodothyronine, which require iodine to make, as well as causing the concentration of iodine in your thyroid gland much higher levels than the rest of your tissues. Glucagon, everybody got for the pancreas. Cortisol, adrenal cortex. Thyroid stimulating hormone, most people got was the pituitary, right? It controls the thyroid gland, but it's not made by the thyroid gland. Choose all that are true about steroids. They're not proteins, right? They're lipids. That's something you've got to know. They're lipids. That's why they need carriers to move around. That's why they can just move through the cell membrane and get into the nucleus without a problem. So most people they need carriers to travel in the bloodstream. They're lipids, and they typically bind with receptors in the nucleus, as opposed to the other kind, like peptide and protein, amino acid based hormones, which bind to receptors that are on the outside, they're on the cell membrane. So, any questions about any of that or anything else that we talked about? And we were discussing endocrine system last class. And then also remember, hormones are used by organs other than the official endocrine glands. The endocrine glands are primarily about releasing hormones, but again, your small intestine releases hormones. Your heart releases hormones. You know, things, so other organs that might not be primarily endocrine organs do have endocrine function, use hormones to communicate with other parts of your body. So don't think of hormones just purely being in the realm of these classic endocrine glands. Okie dokie. So now we are going to start our final topic. It's going to be the reproductive system. So what are functions of the reproductive system?
Can you repeat that? I'm sorry.
Functions, what are the functions?
Babies.
Yeah, to make babies, to reproduce.
So reproduce, so it's to reproduce, make new humans. But there are easier ways to make new, make offspring. Like if you were a sea anemone, you just start kind of budding off a little clone of yourself. And when it got big enough to be independent, it would go off, and it's way more efficient. It's like the way we do it there, most sperm and eggs that get produced never end up becoming a new little human. They just go to waste. There's all of this energy and materials. So why do we go through this and just getting the sperm and the egg together is really kind of this big complicated to do, trying to make sure that happens. What do we gain from this much more kind of complicated, wasteful dance we go through?
Does it also control release of certain hormones? Or
Does it also help balance hormones or regulate hormones?
No, I mean, the hormones, I would say, are more controlling the reproductive system.
Is it to kind of like create diversity in our species?
Yeah, so sexual reproduction. Creates this kind of genetic diversity. Right, so, and it's not just on a physical level, like, we can look around and it's like, oh, this person's taller or shorter, or they got blonde hair or brown hair or red hair, or they got a big nose or a little nose or whatever. But the genetic diversity is down at the level of your enzymes and your membrane proteins and all sorts of things. All the metabolic pathways in each of us works a little bit differently, which is nice. It means that some virus that goes out there, it's probably gonna kill some people, make some people really sick, make some people kind of sick, and some people can barely feel it. So it makes the species as a whole a lot more robust when you got this genetic diversity. And again, make sure when you think about genetic diversity, when you think about these, reuptake pumps that pump serotonin back into your presynaptic terminals, some of us have ones that work faster or slower than others. And it's not like there's a normal version and then some people are outliers. There's like maybe a third of us have one version, a third have another version, a third have another version. Enzymes that are degrading or breaking down things. Some of us have one version that works faster, another half of the population has a version that works slower. And then you've got this crazy deck of all these different things shuffled and dealt out. It makes each of us pretty different, which makes it more or less surprising that different people are going to respond very differently to different drugs, right? You might wonder, why are there so many drugs for the exact same condition? And for some person, this drug is like a miracle and totally takes care of the issue at hand with very few side effects. Yet for another person, the side effects are unbearable and we need to find some other solution, right? So it's useful to remember about genetic diversity, just in terms of just how we're all going to respond kind of differently to the exact same situation or the same drug or whatever. So kind of putting that out there. And the other part of the reproductive system, it's going to be more on the side of the female, is kind of, providing, place, for, embryo or for fetus to grow. So we got to kind of make sperm and eggs and get them together, but then we got to give that little fertilized egg a chance to get a foothold in this world before it kind of goes out and starts becoming an independent little creature. So that's kind of like this big picture. When we talk about kind of the primary sex organs, we are usually just talking about the gonads. Ovaries for a woman, testes for a guy, that are making the gametes. Everything else are accessories. When we're going to talk about the glands that make up the goo and the semen, we're going to talk about the uterus and the fallopian tubes and stuff. And, but at the core, sexual reproduction is going to require the production of gametes. What kind of cell division is going on to create gametes?
Meiosis.
Meiosis. So meiosis, the only place meiosis happens in the body is in the ovaries and the testes. And everything else, all growth and repair and everything is going to be mitosis. I think it's important. We should how is meiosis different from mitosis? This is biology 110.
You end up with haploid cells and 4 daughter cells instead of 2.
Exactly. So mitosis, so both of them start with a single cell and a single 2N cell, a diploid cell. Right, it has 2 copies. There's 23 chromosomes that make up the human blueprint, and each one of your cells has the maternal copy and a paternal copy. So you have 2 alleles for every gene. That should all sound familiar. In mitosis, you basically make copies, sister chromatids of every single chromosome. You pull exact copies of everything to both daughter cells, you're going to end up with two, identical diploid daughter cells. So you're basically just kind of making clones of the original cell that was dividing. Meiosis, you've got two cell divisions going on. There's meiosis 1, meiosis 2. You're going to end up with 4 daughter cells. They're haploid because in the process of metaphase 1, instead of just lining everything up, all the maternal paternal things all along that metaphase plate, you actually pair up the homologous chromosomes and you pull one to one side and one to the other side. And then there's also all sorts of genetic variation that happens. Given the time we've got, I'm not going to go into it, but if you remember, there is independent assortment where either the maternal or paternal one will get pulled to one side or the other side. Also, there was crossing over where the homologous chromosomes are sitting on top of each other and swapping bits and pieces of each other. So you end up with 4 daughter cells. Each daughter cell is only 1 N, and each one has a version of the human genome that has probably never existed before in the universe. Some shuffle of the cards between the ones you got from your mom and your dad. And whether if this is a sperm or an egg, it's going to meet another genetically unique haploid other gamete, and they'll become the 2N zygote that is going to be the beginning of the new little human that's going to have its own unique properties that are going to be mixes of everything. Again, meiosis is happening in the gonads only. It's happening in your ovaries or in your testes. So we can talk a little bit more about the gametes. They're very different. Let's talk about spermatogenesis for a moment. Spermatogenesis is happening obviously in the testes. We'll look at this in more detail. You have these stem cells called spermatogonia. Which are 2N. Yeah, if we were in anatomy, you'd have to learn the names of all these different things. You basically get the primary, hold on, I just spazzed on you here. The first thing that happens is it divides by mitosis and it replaces itself, but then commits another one. It's going to be my primary spermatocyte. This commits into meiosis. Then that goes into meiosis, and there's secondary spermatocytes, there's spermatids. These are the 1N genetically unique things. And then obviously there's going to be this really extensive remodeling because you all know that a sperm doesn't look like a normal cell. So then there is this remodeling called spermiogenesis. Where basically they get rid of most of their cytoplasm and they grow their flagellum. And this is constantly happening. You're constantly having these stem cells, spermatogonia, dividing. They replace themselves because then we're ready to go for the next round. And then we're making these. One normal ejaculation will have like 400 million sperm in it. There's a lot of little dyes. I mean, if if there's less than like 20 million sperm, it's considered getting into infertility, like not enough that are going to be able to make it up to fertilize an egg. There's obviously a flagellum. Flagellum is just like the cilia with that 9+2 arrangement of microtubules. But instead of sweeping things along the surface of some cells, it's actually about making the cells swim. They can swim pretty fast. They can swim like 1 to 4 millimeters a minute. So like if you put one on in a dish, it could probably go a centimeter, almost a centimeter in a minute if it's one of the little fast ones. It's mainly a way to transport genetic material. It's mainly like a nucleus with a little propeller on it. There's this mid piece, what organelle do you think is going to be really important if you're going to have this motor swimming through the reproductive tract? What do you need to run a motor?
Like action potential, right? Or like you need power
You need
somehow.
power, right?
And-
How does that cell make power? How does a cell make energy?
Does it need ATP?
Yeah. How does a cell make most of its ATP?
Mitochondria.
Yeah, exactly. So this is where we find lots of mitochondria. It's actually this kind of interesting spiral mitochondria. So lots of mitochondria to make lots of ATP. So this little, see, we're tying things together. Powerhouse of the cell. It's like, oh yeah. And then there's this kind of bunch of these enzymes at the nose called the acrosome. The reason we need an acrosome is the egg is not ever on its own. The egg is always surrounded by other cells, which I mentioned about this idea of the follicle. So like if here's an ovum, the ovum has all these little cells that are around it. And the sperm are dinky compared to this thing. That acrosome helps start digesting away. Acrosome has enzymes that can digest away, so eventually the sperm can finally get inside and fertilize the egg. Once one sperm actually does manage to get to the egg and send in its nucleus with its genetic material, then that triggers this thing where this membrane starts swelling on the outside of the egg and pushes away all the other sperm. So once one gets in, all the rest get pushed off and pushed away. We call it like blocks to polyspermy. Right, because it's possible to have a viable thing with more, with different genetic mixes, but they're not usually viable. So typically once one gets in, the rest get pushed away. From this picture, you can see the egg is really big. The egg is going to have to survive ovulation, all the way down to getting to the uterus where it can kind of implant and start growing, but it takes about a week. Like once the egg is ovulated and fertilized, it is on its own for about a week. It needs all of its own supplies. It needs to have all its own nutrients and everything it needs to survive until it makes it through the fallopian tubes into the uterus, implants in the wall of the uterus, and starts getting nutrients from the mom. So this ovum, this is like one of the biggest cells in the human body. It's like visible with the naked eye. In oogenesis, which is, the creation of eggs in the body, there are these oogonia, which would be like the spermatogonia, but these are only in the fetus. By the time once you're born, these primary oocytes are committed into meiosis. So that then they go into suspended animation. They just sit there for the next like 10, 13 years until the woman goes through menarche and starts ovulating and having her period and all of that stuff. But so these eggs, though, that when you're born, you have all the eggs you've got, and they've already started meiosis, but then they're stalled out. When they start going through meiosis, they do it a little differently than we saw with the sperm. Each time it splits, you only get one viable egg out of that final process of meiosis, because you want to have all of that cytoplasm and organelles and everything in the final egg, because that thing's got to be big and have all the resources in order to make the journey from the ovary down the fallopian tubes to the uterus, which I said takes about a week or so. So these kind of throwaway things here we call polar bodies. And these are just, they're going to just be non-viable little things that we don't care about. So in oogenesis, it's different from spermatogenesis. One is the oogonia are only in the fetus, and by the time you're born, all of your eggs are already committed into meiosis. And as it completes meiosis, you only end up with one viable, genetically unique haploid cell and 3 little throwaway, these polar body things. So we'll come back to this, because like in addition to the egg, like I said, we actually have these other cells that create this larger structure called the follicle that we're going to follow as well, which is going to be important in this whole process. But in terms of the egg alone, the egg, which is ovum, this is what's going on with it. But again, we're going to look at the related development of the other cells that make this complicated structure that hold the egg. So, all right. So what we're going to do now is we're going to get into detail on both sides here. And, just maybe, what do you call it? The reality of human sexuality is really complicated, and there's whole continuums of, kind of sex and gender and this and that. For the purposes of today, we're going to just make everything simple. We're going to have male and female, males making the sperm, the male gametes, females making the female gametes, the ova, which in reality, you need sperm and eggs to continue the species in general, at least the way we've been doing it up to this point. So this is a useful simplification of this all. So let's dig in here. let's start with the male side, since the male side is simpler. Once we get to the female side, it's going to have to do all the stuff the male side does around making gametes and joining up gametes and all that, as well as, providing a place for the development and growth of the fertilized egg into the embryo and fetus and ultimately baby. So let's start here. Let's start in the testes, which I was saying are the primary sex organs here. We're also going to have to bring in the brain. There's going to be the hypothalamus, the pituitary, and the testes. The word testes, it comes from the same root as testify, to bear witness. Because back in the day, when someone was going to make an oath, they would swear on their, basically their reproductive organs. So that's where that word comes from, testes, to testify, bear witness. So it's all going to start with the hypothalamus. What's the hypothalamus releasing?
Gonadotropin.
Close.
Gonadotropin releasing hormone.
Yeah, gonadotropin releasing hormone, which talks to the pituitary, which then is going to release gonadotropins. Right? And then LH and FSH are going to talk to the testes to either tell it to make more testosterone or make more sperm. So that's what we talked about last class. So we're going to look at that in more detail right now. In the testes, let me make the testes a little bigger so I can draw more in it. It's filled with these tubules, seminiferous tubules, that actually are the place where you're making the sperm. That's going to be listening to FSH. The cells that make up this thing are called Sertoli cells. Sometimes they're called nurse cells because they're helping nurse along the development of the sperm. Those spermatogonia that I was talking about, they're kind of like on the outside of this. They're kind of enveloped. They're not actually inside the Sertoli cells. They're more like enveloped, by these cells. Like think if you had these big weather balloons squeezing you from either side. So here's my little spermatogonia. And meiosis happens. They move, by the time you get into the lumen here, they now have little tails and they are kind of ready to go. So here's our kind of sperm are in the lumen. So meiosis is happening kind of from the outside to the inside of these little seminiferous tubules. The Sertoli cells are actually the target of the FSH. FSH talks to the Sertoli cells. The Sertoli cells then release, I mentioned that word paracrine. They release paracrine factors that stimulate the development of the sperm. So FSH tells the Sertoli cells, release the stuff that you need to release to cause sperm to develop and make more sperm. If there are enough sperm already, there's going to be a negative feedback. So going back, there is this thing called inhibin. The Sertoli cells can send a inhibitory negative feedback thing. If we got enough sperm, it's going to slow down the release of this FSH. So then we're not making as much sperm anymore. So again, we got this negative feedback loop. Other things that are interesting. The walls, these seminiferous tubules have, this is another place where we have like one of these really tight barriers against the bloodstream. Remember we talked about the blood-brain barrier? There is also going to be a blood-testis barrier. That happens for a couple of reasons. One is the sperm that are getting created are so modified from a normal looking body cell, your body would not really recognize it as your own cell anymore and would attack it. So you need to kind of not have it seen by your circulatory system and your antibodies and things like that. Also, in order for sperm to develop normally, it needs to have very high levels of testosterone. There's these special androgen binding proteins in the tubules that hold the testosterone in the lumen at very high concentrations, and you don't want it kind of diffusing back out. So blood testis barrier. Other things about these tubules. Now you've got a lot. You've got, I think, like a half a mile of them in there. So we're going to follow the sperm in a little bit and see what happens to them. But let's talk about the other side first, the LH.
Can I ask a question before we move on?
Sure.
Can you run through inhibin briefly again and where it comes from? I understand that
it's made by the
feedback
Sertoli cells, and it's released by the Sertoli cells if sperm production is already adequate, and it tells the pituitary to slow down.
I have a question too.
Huh?
What was the hormone you mentioned in relation to testosterone, keeping the testosterone in the lumen?
It wasn't a hormone. It was a I said androgen binding protein.
Oh, okay. Thank
So
you.
it's a protein that binds and holds the testosterone in place there.
Thank you.
So LH, which I mentioned, is going to trigger testosterone production. That's going to talk to these other cells in the testes that are making testosterone. They're called the interstitial cells of Leydig. So here in the soft kind of connective tissue of the testes, we're going to have another kind of cell. The interstitial cell of Leydig. Interstitial just means the spaces in between. Leydig's the dude who was describing them. So these make testosterone. And they are going to be triggered by LH. So LH is gonna talk to the interstitial cells to make more testosterone. Remember I mentioned last time, sometimes if you're looking at LH and just thinking about a guy, you can call it interstitial cell stimulating hormone as another name for it. There's also going to be a negative feedback loop here where testosterone will actually work as a negative inhibitory signal to slow down LH production. So testosterone will come back and cause the pituitary to release less LH if there's already enough testosterone. So the one last thing we need to kind of talk about is, well, what about the GnRH, which is pulling the strings on all of this? So gonadotropin releasing hormone in a guy is just pulsed out every few hours. So secreted in pulses. Every few hours. You know, nobody quite knows why it has to be pulsatile, but they have showed that if you just do kind of a chronic infusion of gonadotropin releasing hormone, that it doesn't work right. It has to kind of pulse. It has to kind of go up and down and then up and down, which is then going to talk to the pituitary and cause the pituitary to release its things. These negative feedbacks that I've been talking about seem to work more at the level of the pituitary, making the pituitary less sensitive to the gonadotropin releasing hormone rather than actually working at the GnRH. GnRH can be affected by like psychic factors, just your brain. There's like one of the classic things like the sailors that have been out at sea and are getting near port and are going to finally see their sweeties after 6 months and are getting all excited. Their gonadotropin releasing hormone starts going up more than normal. They get more LH than normal, more testosterone than normal. Their beards start growing faster than normal. So psychic factors can also affect gonadotropin releasing hormone. But in general, it's just constantly pulsing out, which is going to be super different than what we find in the woman. In the woman, it's going to go up at the beginning of the month and then get suppressed and come down for the next 30 days and then come back up to start the next cycle. So it's going to be very different when we get to the female reproductive system. All right. We are now going to add in, this is kind of like the primary sex organ, but now obviously sperm alone need to get mixed up with lots of other things to become semen and need to go at the penis and go find their egg to meet up with and all that. So we're gonna put this all together in a bigger picture. All right. This is supposed to be kind of the abdominopelvic cavity here. There's a bladder. The urinary bladder, I should say. The urethra is going to come down and will come out. There'll be a penis. And obviously the testes are hanging outside the body in the scrotum. Usually it's pubic hair on there. Obviously there's another one on the other side, just for make this more clear. We'll have this guy have one testis here. So why are the testes, which are so critical for the propagation of the human species, hanging outside the body in a little skin bag in this really vulnerable place? Why not have them more protected? You know, like the ovaries are deep in your pelvic cavity.
Does it have to do
Is
with
it for temperature control?
Say that again.
what she said, temperature?
Yeah, so for spermatogenesis to occur, they have to be a few degrees cooler than normal body temperature if spermatogenesis is going to proceed properly. So there's outside so they can be cooler. There's muscles. Again, there's a cremaster muscle actually, which is connected to your abdominal wall. which can pull the whole shebang up closer to your body to keep it warmer, or relax and let things dangle for more airflow if you want to keep things cooler. Cremaster muscle is also activated during sympathetic. Remember, we talked about sympathetic activation. Your eyes dilate and your bronchial passages open up and your heart speeds up. Another thing that happens if you're a dude is your cremaster muscle contracts and your testes get pulled up out of danger as much as possible. So it's activated by the sympathetic nervous system as well. There's another muscle, the dartos muscle, more just in the skin, which can make things more wrinkled or less wrinkled and smooth to increase or decrease surface area for heat control. So you've got a couple of muscles. You got the one in the skin that makes things wrinkled or not. You've got the cremaster muscle that pulls things up towards the body or not to kind of adjust temperature. The sperm obviously are getting made in these little tubules in here. They kind of go to this area, kind of the rete testes. Again, for this class, you don't need to know the details. They leave the testes and they end up in another tube, in this big tube called the epididymis. Which is kind of storage and maturation of the sperm. When a sperm is created and stored, it is in suspended animation, right? It'd be a waste of energy if your sperm were swimming and bouncing off the walls and just getting ready to go. And they're just, unless they're actually ejaculated and on their way, they are going to be in this state of suspended animation. When we talk about semen, part of what is going to be in semen is going to be stuff to what we call capacitate the sperm, kind of wake them up. Like a sperm that is just sitting at this stage is not awake. It's not ready to just swim and fertilize something. It needs certain factors to wake it up. Then, obviously, the sperm are going to ultimately have to come out the tip of the penis. So they got to get up into the urethra. So they're going to do that through this tube called the Vas Deferens. Also called the ductus deferens. It gets into your body through a little tunnel. That little tunnel is called the inguinal canal. We're going to see when we get to the female reproductive tract, there is a whole lot of what we call homologies between the male and female. Like up to the first, I think, a few months, both male and female look exactly the same. In the embryo, whether it's going to be a male or a female, ultimately, it's got all the same parts and pieces, internal and external. It's not until these levels of testosterone start going up during development that things start changing. But like what ends up being the scrotal sac in the guy is the labia majora in the woman. What ends up being folding, the shaft of the penis in the guy is the labia minora. But in the guy, those parts actually fold around and envelop the urethra to create the shaft of the penis. In a woman, there's actually the round ligament that actually goes through her inguinal canal and anchors in her labia majora. So it's kind of fascinating when you look at the homologies, the ways they're similar. We'll see more of those as we go on. Like the tip of the penis And so the glans penis and the glans clitoris, start as the same thing, and ultimately, play the same role in a lot of ways. They're all full of nerve, kind of receptors and nerve endings for sexual pleasure. So vas deferens, there's also this going to be obviously coming through the inguinal canal. There's going to have to be arteries bringing blood down, veins bringing blood back up. So everything together is called the spermatic cord. The spermatic cord is just it's the vas deferens as well as the testicular arteries and veins that are all serving, and nerves as well. Obviously, there are afferent signals coming from the testes causing agonizing pain and nausea if they've been kind of injured. One of the cool things I'll mention really briefly, because it's just cool. The testicular artery comes down, bringing blood down into the testes. The veins that are returning wrap around the artery in this kind of network. So that we call it the pampiniform plexus. Envelop the testicular artery. Not there. So basically, as the warm blood is coming from the body, it's getting pre-cooled by this blood that's already been hanging outside the body cooling down. So it's this way to kind of pre-cool the blood that's entering down into the testes. Can you see that from that picture? It's pretty cool. All right, so obviously the sperm are just one part of the ejaculate. In fact, they are only maybe 10% of what ends up being a guy's semen. So the testes make maybe 10% of the ejaculate. The rest is being made by two different glands. There is going to be the seminiferous gland. Let me give it a good color. Oh, give it a different color. I'll make it yellow. It's actually kind of on the back of the bladder, but I'll just put it here on the front to make it easier to see. The seminiferous gland is making maybe 60% of the semen. Granted, semen is that goo that the guy ejaculates. This is going to include things like, fructose. Give the little dudes some energy so they can make ATP and swim. It's going to include fibrinogen. When a guy ejaculates, you actually have that clotting, that protein will actually clot. It'll kind of gellify the semen, so you don't want to squirt it up the female reproductive tract and just have it pour right out. So once it gets kind of ejaculated up, it actually kind of jellifies. So it kind of sticks up in there, and then it kind of can reliquefy and the little dudes can keep going on their way. The other gland that is making semen is going to be what's called the prostate gland. The prostate gland actually envelops around the urethra. And then it has little ducts that feed into the urethra. But you can kind of think of it like a walnut with a hole drilled out the middle. This has a variety of things. One of the most important things, helps capacitate the sperm. Again, capacitate is that word I was saying, kind of wakes them up. It's how they get all out of their torpor and start swimming and become capable of fertilizing an egg. When a sperm is first ejaculated, even if it was at the egg immediately, it would take it like 1 to 9 hours before it could actually do its thing and get and fertilize. It takes them a while to get fully activated. Once they're out, they can live for quite a while. They can live at least a few days, up to 5 days even. So you can actually get pregnant you can have sex 5 days before you ovulate, and there's a chance that there's some sperm still swimming around that's viable, that once the egg becomes, kind of pops out of the ovary, that sperm is like, boom, is there. So the sperm, the window of getting pregnant can be from sperm that might have got deposited 5 days before you've ovulated even. Typically, it's more like 1 to 3 days for these little dudes to live. What else? So in terms of, there's one other gland we need to add in, the bulbourethral gland or Cowper's gland, which is not part of the semen, but is going to be to help kind of clean out the urethra.
How do you say that word?
Bulbourethral. So like when a guy is getting sexually excited and it's likely that there's going to be an ejaculation, the urethra is where you've had urine coming out. So these bulbourethral glands make this kind of mucousy secretion that kind of clear out the lines. It's that precum, that kind of slippery mucus drop that ends up at the tip of the dude's penis as he's getting aroused. So that kind of precum, usually what people call it. So that's kind of before ejaculation. And a woman is going to have a homologous gland, the Bartholin's glands that empty into right near the entrance to the vagina. So let's talk about ejaculation. If you remember, we had talked about the urethra having an internal sphincter and an external sphincter. So in this picture, let's add them in. Here and here. So when a guy is getting aroused, we'll talk about this in more detail. Then we have this more parasympathetic thing that's resulting in erection and all of that. But at the climax, there's going to be this thing happening, it's ejaculation where the semen mixes up and gets squirted out. These sphincters close down and block off this little section of the urethra. So what we call the prostatic urethra there. And at that point, you have 3 things mixing in. You've got the testes. Testes are maybe, in fact, let me just go on another one. So ejaculation, first is going to be what we call the emission phase. This is kind of the mixing. Like 10% of it is kind of from the testes. It's going to be the sperm and the fluid coming from there. 60% from the seminiferous tubule, from the seminiferous gland. Not seminal gland. Oh my God. 30% from the prostate gland. So at this point, they're all mixing up in here, but it's kind of just swelling out this whole thing, and they're all coming together. And then this external sphincter, so seminal gland, not seminiferous gland. They're all mixing up. External sphincter relaxes, and now they can make it out the penile or spongy urethra and get ejaculated. So that part is called the expulsion phase. There are muscles at the base of the penis that can contract. I did the calculation of this. So if they can get squirted out somewhere like 11 or 12 miles an hour. So this is that white goo, that semen, which is mainly not sperm, right? Only 10% of the stuff is coming from the testes. In male sterilization, you can go and you can just cut the vas deferens. Well, maybe I do it red. Right, you can snip the vas deferens so the sperm never make it up into the mix. What's it going to look like when the dude ejaculates if he's had a vasectomy?
Probably exactly the same.
It's going to still be white goo. It's the only way you're going to know anything's different if you look under the microscope, and you'll realize that there's no sperm swimming around in there. So let's talk. Last thing we need to talk about is erection. Obviously, the penis is not going to be able to get up into the vagina well if it's just flopping there. So there is penile tumescence, the erection. This is like not a very pretty penis here. Let's try it again. Back. Okay, so penis. Yeah, so there's the glans penis. Glans actually means acorn, especially if the guy still has the prepuce or the foreskin. You can kind of see why it's called like an acorn. This is where you have a majority of like the little nerves that are, for sexual pleasure. And like I was saying, the glans penis is homologous to the glans clitoris. In the woman, there's obviously the urethra coming through, and then there's the spongy tissue. Spongy tissue, there's the corpus spongiosum, the corpus cavernosum. And spongy tissue is kind of what it sounds like. It's got lots of spaces in between that can fill and engorge with blood, and that's what's going to cause the penile tumescence, kind of swelling up and becoming erect. So during sexual excitement, during arousal, you increase the blood flow into here, you decrease the amount of outflow, and that's basically kind of blowing it all up and swelling it up. The corpus cavernosa have what are called the crura. These kind of anchor on the abdominal, kind of on the pelvis, basically. So as the penis gets erect, it's not just going to get stiff, but it will also kind of become erect with respect to your body, instead of just flopping around like a erect thing and hanging off the front of you. So spongy tissue filling with blood due to vasodilation, more blood coming in, and then usually keeping the blood from flowing out. So you get this kind of inflation going on. This is going to be an important part of just overall sexual response in both male and females. We could say vasocongestion. In a woman during sexual arousal, there is spongy tissue in the clitoris, and the clitoris is also engorging with blood and becoming more sensitive, as well as just in the whole groin area. Actually, the uterus ends up kind of folding back and stretching out and elongating the vaginal canal. It's like there's a lot of stuff going on in a woman during sexual arousal. It's just not as obvious to see because, you know, obviously a guy's got the penis hanging out in front and you can see it swelling and getting erect, but there's a lot of stuff going on in the woman as well during arousal. And a lot of it is resulting from this vasocongestion, kind of more blood coming into the region and swelling up structures. So we'll talk a little more about that. So let's see. I think for the guy, that's about it. There is, obviously there's more things we could talk about, but in general, you know, the one thing I can't talk, I'll mention one more thing, just testosterone levels. You know, in a woman, there's a more dramatic menopause thing that happens, like sometime in 40s or 50s, the ovaries will start kind of shutting down. Again, those eggs that have been waiting to go since she was born are finally kind of done. But in a guy, you don't have a dramatic thing like that, but you still, and the guy is making sperm till the very end, but there is also a decline. Testosterone levels, like if you're like, you know, 18 years old versus 50 years old versus 80 years old, it goes down. By the time someone's in their 80s, it's going to be way less than half probably of what it was when they were really young. So there is an overall decrease in testosterone as a guy gets older. So I'll put that out there.